Domain databases are resources that provide information about protein and nucleic acid domains, including their sequences, structures, and functional annotations. These databases are essential for researchers in understanding protein function, predicting interactions, and exploring evolutionary relationships. Domain analysis databases provide essential tools for identifying and analyzing protein domains, similar to motif finding tools used for sequence motifs. Notable databases include PFAM, which offers a comprehensive collection of protein families and domains, utilizing multiple sequence alignments and hidden Markov models for accurate domain identification. SMART focuses on the identification and annotation of protein domains involved in signaling and interactions, allowing users to visualize domain architectures. UniProt integrates various protein signature databases, including PFAM and SMART, to provide a unified platform for domain identification and functional annotations. Additionally, CATH and SCOP classify proteins based on their structural features and evolutionary relationships, facilitating exploration of protein structures and their domain compositions. ProSite offers a database of protein families and functional sites, providing patterns and profiles for identifying functional domains. For structural comparison, DALI allows users to analyze protein domains by comparing their three-dimensional structures, while Gene3D focuses on protein domain identification based on structural data. These tools collectively aid researchers in understanding protein function, structure, and evolution, making them invaluable resources in molecular biology and bioinformatics. How to identify that our sequence downloaded from UniProt is the right one and that they contain the domain that we searched for that, you need to go to Motif Finder database. Search Motif Finder in the Google and click on the first website.
To find protein domains using a motif database, such as the one provided by KEGG, Kyoto Encyclopedia of Genes and Genomes, you can follow a series of steps. First, access the database website and prepare your protein or nucleotide sequence, ensuring it is in the correct format, typically FASTA. In the database, locate the Search or Input section, where you can paste your sequence or upload a file. Depending on the database, you may have options to set specific search parameters, such as the type of analysis, protein or DNA motifs, E-value thresholds, and database selection for motif matching. After entering your sequence and adjusting the parameters, initiate the search by clicking the Submit button. Once the search is complete, review the results to see identified motifs or domains present in your sequence, along with links to detailed information about each domain, including functional annotations and structural data. You can analyze these domains by exploring their biological functions, structural information, and evolutionary insights. Some databases also offer options to export the results for further analysis. For effective domain identification, ensure your input sequences are accurate, consult the database's documentation for detailed instructions, and consider cross-referencing results with multiple databases for a comprehensive analysis. This process can yield valuable insights into the functional roles of proteins and their evolutionary relationships, making motif databases essential tools in bioinformatics and molecular biology research. To identify protein domains using the Motif Finder tool from the KEGG website, start by visiting the Motif Finder page. Input your protein sequence in the text area provided, either by pasting it in FASTA format, beginning with a header line, e.g., protein_name, followed by the amino acid sequence, or by uploading a FASTA file directly from your computer using the Choose File button. After entering your sequence, select the relevant search parameters, ensuring that the input format is set to FASTA. You can choose the default search settings or specify custom parameters for the motifs you wish to identify. Once you are ready, click the Submit button to initiate the motif search. The tool will process your input and analyze the sequence for known motifs. After the search is complete, the results page will display any identified motifs along with their corresponding domains and annotations. Each motif will typically link to additional information, including its description, functional annotations, and any related pathways or proteins. Review the position of motifs within your sequence to understand their biological relevance, and if needed, Utilize the download option to save your results for further analysis in a preferred format such as CSV or TXT. Finally, use the identified motifs and domains to enhance your research, whether you are studying protein function, structure, or interactions, incorporating these findings into your projects or papers. In the upper part, there are two sequences in the box. Upper one is query sequence YABI domain and lower one is the YABI domain sequences from the database. The database domain ID is also listed above StatRed with P. Highlight the red sequences. This is the YABI domain sequences from the query sequences that you downloaded from UniProt. Then we will paste this sequence to the already downloaded file from UniProt and will keep this domain sequence for record for future use. We will also copy the datab's YABI domain sequence from the database and will paste in the same file for future use. F. This ID is the peptide ID sequences from DAM database. A Motif Finder is a sophisticated computational tool widely utilized in the field of bioinformatics for the identification of motifs within biological sequences. Motifs are short, recurring patterns in DNA, RNA, and protein sequences that are crucial for understanding the functional aspects of these biological molecules. In particular, motifs often signify important structural or functional roles, acting as building blocks for the organization and functionality of proteins. Thus, the identification of these motifs aids in uncovering conserved sequences that may play vital roles in various biological processes. The significance of motif finding lies in its ability to reveal conserved patterns across different species. These conserved motifs can indicate regions of sequence that have remained stable throughout evolution due to their critical functional importance. By employing motif finders, researchers can delineate potential protein domains that are associated with specific biological functions or interactions, thus enhancing our comprehension of protein behavior and interactions within cellular contexts. Understanding these motifs enables scientists to make inferences about protein function and suggests possible implications in areas such as disease mechanisms and evolutionary biology. Furthermore, motif finding is integral in the context of genome-wide analysis, where vast quantities of sequence data are examined. The application of motif finders can streamline the process of searching for biologically relevant sequences, significantly improving the efficiency of bioinformatics analyses. These tools often integrate various algorithms designed to detect varied types of motifs, which can increase the resolution and accuracy of the findings. The insights garnered from motif identification, consequently, contribute vastly to our understanding of gene regulation, molecular interactions, and evolutionary conservation, thereby emphasizing the importance of motif finders in modern biological research. Motif finders play a crucial role in genome-wide analysis, providing substantial insights into the functional elements of genomes. One of the primary applications of motif finding tools is the annotation of protein domains. By identifying conserved sequences within protein structures, researchers can predict the function of genes and their products, ultimately shedding light on biological processes. This annotation is vital for understanding how proteins interact with one another and with various cellular components. Furthermore, motif finders assist in elucidating gene regulation mechanisms. Regulatory motifs, often located in promoter regions or enhancers, are fundamental for the proper expression of genes. By utilizing motif finding techniques, scientists can discover transcription factor binding sites and study their influence on gene expression. Understanding these regulatory networks is essential for dissecting complex biological pathways and developing therapeutic strategies targeting specific genes. Another significant application of motif finders is the discovery of new protein families. As researchers sequence more genomes, the need for identifying previously unknown protein domains becomes increasingly important. Motif finders can reveal novel conserved sequences across diverse species, suggesting evolutionary relationships and potential functional roles. Such discoveries can lead to advancements in areas like evolutionary biology and functional genomics. Comparative genomics is yet another field where motif finding demonstrates its utility. By analyzing motifs across different species, researchers can identify conserved elements that may indicate essential biological functions. Case studies exemplifying this application include evolutionary studies of specific gene families or pathways, where motif analysis uncovers conservation patterns that highlight critical evolutionary events. Overall, the applications of motif finders in genome-wide analysis are diverse and impactful, playing a pivotal role in advancing our understanding of molecular biology and genetics. To copy and paste a sequence in FASTA format for future bioinformatics analysis, follow these simple steps. Step-by-step guide. Open a text editor. Open a plain text editor on your computer. Common options include Notepad, Windows, TextEdit, Mac, in plain text mode, or any code editor like Visual Studio Code or Sublime Text. Format your sequence in FASTA format. The FASTA format requires each sequence to start with a header line, which begins with a symbol followed by a descriptive name or identifier for the sequence. The subsequent lines contain the actual nucleotide or protein sequence. Ensure that the sequence is continuous without spaces. If the sequence is long, you can break it into multiple lines, but do not insert any spaces or special characters. Here's an example of how to format it. Copy the sequence. If your sequence is already in a source, like a website or database, select the entire sequence, including the header, and copy it, Ctrl+C on Windows or Command+C on Mac. Paste into the text editor. Click into your open text editor and paste the copied sequence, Ctrl+V on Windows or Command+V on Mac. Ensure that it appears correctly formatted. Save the file. After pasting the sequence, save the file. Click on File in the menu and select Save As. Choose a suitable location on your computer. Name your file with a .fasta or .fa extension, e.g., yabb1.fasta. Make sure to select All Files in the File Type dropdown if using Notepad, so it doesn't save as a .txt file. Verify the file format. After saving, you can reopen the file in your text editor to ensure it retains the correct FASTA format. The header should remain intact, and the sequence should follow it properly. By following these steps, you'll create a FASTA file containing your protein or nucleotide sequence, ready for use in bioinformatics analysis. This file can then be used in various software tools for sequence alignment, motif searching, or other analyses as needed.